All right. Well, I'd like to welcome everyone to today's Polyploidy webinar. Today we have what looks like two really exciting talks. And up first is Maya Wilson-Brown. Maya's a fourth-year PhD student in Dr. Emily Joseph's lab at Michigan State University in the Department of Plant Biology. Her dissertation research is focused on the impact of interploidy gene flow on the evolution of Capsella bursa-pastoris. And today she's going to tell us about her work on gene flow between ploidies and variation in Capsella bursa-pastoris. I'll let Maya take it away. Thank you.
Thank you all so much for being here. This is kind of like a fun webinar to do because I don't frequently talk to other polyploid specific scientists. But I did want to start with a quote that I hope that we can all appreciate. And this is a quote I love from a review by Dr. Sally Otto. That says that polyploidy is a massive genomic mutation. Polyploidization represents one of the most dramatic mutations known to occur. And we know this is true because polyploidy is associated with a lot of genomic activity, often including proliferation of transposable elements, gene, chromosome, and even sometimes genome loss. It can make cell division really complicated. And then on top of all that, for newly formed polyploids, there are also extrinsic difficulties in establishing because of competition, either sexual competition or ecological competition with related majority cytotypes. But the quote goes on to say that polyploidy is well tolerated by many groups of eukaryotes. And like Dr. Barker said, I'm in a plant biology department, so I tend to think about polyploid plants most often, and particularly angiosperms. Um, so I'm a little bit biased, and I do think that plants are the best at tolerating excessive numbers of chromosomes. So I apologize that all of my examples are in plants. But I think this is sort of the question that unites all of us as biologists, and particularly those of us who are interested in evolution, because if polyploidy is so difficult, then why are there so many polyploids? And it drives a lot of our research and our curiosity. And because of this, there are a lot of ways to think about this question. Um, Sometimes we can consider how taxonomy and evolutionary histories might influence the prevalence of polyploids, including rates of formation, diversification, and loss of polyploid lineages. We can think about the ways in which a polyploid might have formed from the spectrum between autopolyploidy to allopolyploidy. Um, uh, in plants, we have this beautiful array of mating systems, including outcrossing, selfing, clonal reproduction, and even apomixis. We have genetic and genomic interactions, as well as ecological changes. And on top of all of that, all of these things interact, of course. So I think that it's these really complex interactions that contribute to a lack of super strong support for any one of these features in explaining the prevalence of and patterns of polyploids across the tree of life. So that's all just to say that polyploids are really diverse. And so the relative impact of any one of these features depends on the system and its evolutionary context. Okay. So I think that there is another aspect of polyploid evolution that is often overlooked, and that is gene flow between ploidies. And I think gene flow between ploidies could have a really significant impact on our understanding of polyploid evolution, because it means that the gametes of newly formed polyploids are not wasted, which allows for population growth. It might increase genetic diversity, which is important for evolution and population resilience. And it also allows for the possibility of adaptive introgression entering a population at really high frequencies, which could speed up adaptation. Um, and there are a lot of historical and practical reasons, I think, for interploid gene flow being understudied. And that's not to say that it hasn't been studied at all. I think we have actually a lot of empirical evidence of gene flow between ploidies, but it's just been difficult to get at until like recent years. And so last year, this review was published on the evolutionary significance of interploid gene flow. And they say that the genomics era provides a novel opportunity to reevaluate the role of interploidy introgression in speciation and adaptation. And to achieve this goal, interdisciplinary studies bordering ecology and population genetics and genomics are needed. I'm not going to touch on all of these things in this talk, or maybe even ever, but I do want to introduce you to a little bit of what I've been exploring regarding interploid introgression in a natural system, because I do think that there is now the potential to explore all of these things in a synergistic way. Okay, so all of my work is within the genus Capsella. Capsella is in the family Brassicaceae, and it's pretty closely related to Arabidopsis. There are 4 recognized species in the genus: Capsella orientalis, Capsella bursa-pastoris, Capsella rubella, and Capsella grandiflora. Capsella orientalis, rubella, and grandiflora are all diploid, while Capsella bursa-pastoris is an allotetraploid. So it has 2 diploid subgenomes that were derived from a Capsella orientalis-like ancestor and a Capsella grandiflora-like ancestor. And out of the 4 species, Capsella bursa-pastoris has the broadest geographic and ecological distribution. I think these are points from GBIF. And we think it originated somewhere around the Mediterranean and has subsequently spread throughout Eurasia and then to other continents through human-mediated dispersal. It also has quite substantial variation in leaf shapes, which another graduate student in the lab works on, as well as flowering time. So I think it's like a really great system to try to understand how polyploids adapt to new environments, as well as the recency of its introduction to other continents is interesting in terms of evolution. Okay, so previous studies were looking at the population structure of Capsella bursa-pastoris in its ancestral range of Eurasia. And so on the right, I'm showing results from Corneal et al. In 2015. They were using SNP data to look at population structure in the ancestral range, and they found about 3-ish genetic groups. And then again in 2019, there was an effort to look at global population structure of Capsella from Wesley et al. In 2019, and they found about 2-ish populations worldwide, although I will note that they didn't have samples from Eastern Asia that the Corneal study did. And so for a while, the Capsella community was aligning bursa-pastoris genetic data to the Capsella rubella genome because it was available. And so I wanted to start working on a Capsella bursa-pastoris reference genome, because if we're interested in diversity, we might be missing some by mapping reads to a genome from a different species that has presumably half the genomic material that bursa-pastoris does. And so what we did was we developed a reference genome and then mapped publicly available Capsella bursa-pastoris reads to that Capsella bursa-pastoris reference genome, which was developed by a bursa-pastoris individual from New York City. So first, we just wanted to see like what kind of population structure we can see in the ancestral range when mapped to the new reference genome. And we recover these like 3 ancestral groups like other groups did, 1 in Northern Eurasia, 1 in Eastern Asia, and then 1 around the Mediterranean, which is all well and good. It's always good to have things that align with previous research. And then we also wanted to look at Capsella bursa-pastoris in its introduced range in the United States. And so when we add US populations to the PCA, we see that US populations are most similar to the Mediterranean and the Northern Eurasian groups. And like the Wesley et al. Paper, we didn't detect any individuals that were similar to the East Asian group in the United States even though we had East Asian individuals from the ancestral range in our sample set. Okay, so that's great and well and good. But why do we care about this population structure at all? And I think the reason it's significant is because this population structure is associated with a variation in history of introgression. So there's been a lot of previous work showing evidence of introgression in Capsella bursa-pastoris in the ancestral range that varies geographically. So first in 2008, there was evidence of non-zero migration rates from Capsella rubella into Capsella bursa-pastoris in Western Eurasia, but not in Eastern Eurasia. And then again in 2015, an admixture analysis showed introgression from Capsella rubella in Western Eurasian, Northwestern Chinese populations, but not Eastern Chinese populations. And then again in 2019, an ABBA test showed something similar with evidence of gene flow from Capsella rubella into European and Mediterranean Capsella bursa-pastoris. And so one question we can ask, given the similarity of US Capsella bursa-pastoris to Northern Eurasian and Mediterranean bursa-pastoris, is whether or not we can detect introgression in these US populations. For this portion of the study, I'm just looking at individuals from New York City, which are shown in the upper left. That pink blob is mostly individuals from New York City. And because we have whole genome sequencing and a reference genome, we can also ask not just if there's evidence of introgression or not, but which loci are actually introgressed and if there is variation for introgression among these populations. So to do this, I used Ancestry HMM to perform a local ancestry inference. This method basically gives you a probability for ancestry at specific loci, as opposed to something like admixture that tells you the relative proportion of ancestry within an individual. And it does this by taking the allele frequencies from the parental populations and comparing them to allele frequencies in an admixed population. So for this study, I'm trying to distinguish Capsella rubella ancestry from Capsella bursa-pastoris ancestry. So for my parental populations, I used Capsella rubella and this East Asian Capsella bursa-pastoris that doesn't appear to have a history of introgression with Capsella rubella. And then for my queried population, I'm using Capsella bursa-pastoris from New York City, as well as the Northern Eurasia and the Mediterranean. Okay, so we define allele frequencies at highly differentiated SNPs in these parental populations. And then we compare the allele frequencies in the admixed population to these parental allele frequencies. Oops. Cool. And then we get a probability of parental ancestry at each SNP. So the next few plots I'm going to show you will have bars representing a set of homologous chromosomes for individuals on the x-axis and position along the y-axis. And green color will represent homozygous Capsella bursa-pastoris ancestry, while pink will be Capsella rubella ancestry. And then any gray regions are inferred to be homozygous, or heterozygous, sorry. And so when we look at New York City, this is one subgenome of individuals, Capsella bursa-pastoris individuals from New York City. We do have evidence of introgression, as you can see by the pink bars, from Capsella rubella, which is in alignment with our population clustering analysis, since New York City is so closely related to that Northern Eurasian population. Then for this next plot, you can imagine that I've rotated the previous plot 90 degrees and then stacked individuals on top of one another. So that position is now on the x-axis and different chromosomes are on the y-axis. And introgressed regions across individuals are shown in purple, where darker shades of purple indicate that more individuals share that region as introgressed. And so here we can see that in New York City, there is quite a bit of variation Some regions are very, very common across and fixed across all 60 individuals in New York City. And the introgressed regions vary in length quite a bit. But there are some regions that are polymorphic within this population. So in comparison to introgression in populations from the ancestral range, we can see even more variation, but a lot of the same regions are polymorphic across all of the populations. So I think together, the patterns in the New York City population on its own and in comparison to populations in the ancestral range suggest that introgression from Capsella rubella is certainly older than the introduction to New York City, which we would expect because Capsella rubella does not exist in the United States. And that gene flow from Capsella rubella happened potentially in multiple pulses. Okay. You also might recall that I used Capsella bursa-pastoris individuals in order to define Capsella bursa-pastoris ancestry. And so I wanted to verify that there was no Capsella rubella ancestry within those individuals. So every time I ran the analysis, I withheld a random sample of 5 East Asian Capsella bursa-pastoris individuals from that parental population and evaluated them along with the mixed population. And so here I'm just checking to see that I don't infer any Capsella rubella ancestry in these individuals, and I don't, which is great. Okay, and then the last thing is I wanted to make sure that the signals that Ancestry HMM was picking up were actual Capsella rubella ancestry as opposed to ancestry from another Capsella species or something else in terms of like odd allele frequencies. And so I use LowStruct to perform principal component analysis in windows along the genome, so I could visualize how closely these two methods align. So for example, in this window, we have no individuals that have Capsella rubella inferred ancestry. And then when we look at the PCA for that window, we can see that that is true. So Capsella rubella is shown in the pink triangles, and none of the Capsella bursa-pastoris individuals cluster with Capsella rubella there. That's in contrast to a window where we have no New York City individuals showing Capsella rubella introgression, but a handful of Northern Eurasian and Mediterranean individuals do. And that is also supported by a PCA for that window where we have some Northern Eurasian and some Mediterranean individuals clustering with Capsella rubella and no New York City individuals clustering there. It's kind of hard to see them, but they are like back there in the bottom right corner. Okay. And then finally, I wanted to evaluate nucleotide diversity. So I looked in 100 kilobase per windows across the genome for ancestral populations as well as New York City. And we can see that average pi is quite a bit higher in the introgressed populations compared to the East Asian population, which does not appear to have introgression. Okay, so all of this to say that we have evidence of introgression from Capsella rubella in ancestral Northern Eurasian and Mediterranean populations of bursa-pastoris. The US populations of bursa-pastoris are most similar to these Northern Eurasian and Mediterranean ancestral populations. Genomic regions inferred to be introgressed vary across these populations and within New York City, but the fact that these introgressed regions are maintained even after introduction to the United States, along with the fact that introgression does seem to be associated with increased nucleotide diversity, might possibly suggest an adaptive role for introgression in this system. Okay. And with that, I would like to thank my current lab members, as well as previous lab members, the MSU Genomics Core, ICER, the Institute for Cyber-Enabled Research at MSU, as well as my funding sources, and I would be happy to take questions.
All right. Well, thank you so much. That was really exciting. if you have any questions, folks, please feel free to raise your hand or put them in the chat. And usually it takes a couple seconds. Well, I can ask a question real quick that I have. This is really interesting. So you have a sense of, or any plans to look at the demographic sort of picture here to understand maybe when and these integressions have happened and place them in sort of context? I was sort of curious about that.
Yeah, definitely. I think one of the things I'm interested in is understanding when and where exactly this introgression happened and what is the relationship between introgression in the Mediterranean population and the Northern Eurasian population. I think partially because that might provide more evidence for if some of these introgressed regions are adaptive in certain contexts. And we don't currently see, at least not in the PCAs, like admixture between the Northern Eurasian and Mediterranean populations, but that doesn't mean that it's not happening. And so that could be another avenue for just generating a lot of diversity within bursa-pastoris, especially the introgressed.
And I was also trying to look at how many, it seems like the integration regions are, they're little blocks, of course. So I was trying to sort of get a sense of has it happened? Did it happen a while ago and then they've been broken up by recombination, or they're little? Is it? I'm just trying to get a sense of from that picture. I don't know what the haplotype, sort of what block sizes are like in Capsella. I assume because there's a lot of inbreeding, they must be pretty large in general. So then, anyway, I was just trying to get an impression of it. How's that?
Yeah, for sure. I definitely think like there is a lot of variation in size, and so that to me indicates that is maybe a part of that multiple pulse hypothesis. But it's also hard to say. I mean, like, I think that the polyploidy does add a layer of complexity to this, which is actually really exciting. So
no, that's really wonderful. Well, thank you. Are there any other questions for Maya at this time? All right. Well, if not, we'll have a chance at the end to ask Maya questions if you have any that you want to come back to and ask them. But we'll go ahead and ask Nomar to start sharing his screen and move on to our second talk of the day. But thank you, Maya. That was really, really, really cool.
I hope you can see me and you can hear me.
Yes, absolutely. All right. Well, our next speaker is Nomar Waminal. Nomar is currently a postdoc at the Leibniz Institute of Plant Genetics and Crop Plant Research. He is an evolutionary biologist specializing in plant cytophylogenomics and employs cytogenomic approaches to understand karyotype evolution and its influence on speciation. And today, Nomar is going to tell us about the Crocus panrepeatome, duplications, dynamics, and dysploidy. Thank you, Nomar.
Thank you, Mike, for that kind introduction. And good morning, everyone. My background is in cytogenetics, and my talk today, we're trying to understand the relationships between whole genome duplications and how these promote repeat bursts, and how these dynamics of repeats influence dysploidy, change in chromosome numbers, or dysploidy. And we are using Crocus to study this mechanism or this process. And to do that, we need to we analyze the panrepeatome, or the total repeats in the different several genomes in Crocus. Wait a moment, maybe I. Okay. We know that angiosperms share not just the flowers, but at least one round of whole genome duplications. And whole genome duplications is known to have spurred the diversification of the angiosperms. However, whole genome duplication per se does not cause diversification, but what follows after whole genome duplication, or the diploidization processes, which could include repeat bursts after this genomic shock, transcriptomic shock. And I think Maya mentioned that in her talk as well, that this could influence the epigenetics and the expansion of repeats. And repeats could also increase the chance of aberrant chromosomal rearrangements when repeats are found in two different chromosomes, and these two double-strand breaks are misrepaired ectopically and will result to different chromosomes, or it could lower the chromosome number, or it could increase the chromosome number in the process called dysploidy. And when these resulting lines can have reduced recombination because of dysploidy, this will eventually result in reproductive isolation and then speciation. And this cycle of karyotype evolution could continue and could help explain why, despite of the different rounds of whole genome duplications in different lines in angiosperms, we could still see a low chromosome count of basic chromosome number between 7 to 12. So in this process, this whole genome duplication, repeat burst, diploidization or dysploidy, and reproductive isolation, we term this a karyotype evolution cycle, which could continue in different lineages. But I want to focus particularly in dysploidy, which is relevant to my talk today. But in order to understand dysploidy, first we need to understand the structure or the landscape of a chromosome. This is essentially the bird's eye view, or the general organization, where we have repeats, because repeats play a significant role in these chromosomal rearrangements. And repeats could either be categorized based on their location or their organization. Based on organization, they could be either tandem repeats or dispersed repeats. Tandem repeats are arranged from in a head-to-tail fashion like this, while dispersed repeats are dispersed, scattered without any particular pattern. And these include the transposable elements, while tandem repeats include the many satellites, and they are often organized in specific loci in the chromosomes. And we have here the telomeric repeats. We know these are highly repetitive regions in the chromosomes. The subtelomeric repeats, and they could also be found, the satellite repeats, interstitially in the chromosomes, in the pericentromere, or in the centromeric repeat. Now, with this idea, I'm showing you this one of the mechanisms. There are several explanations to descending dysploidy. And one of these is called the end-to-end translocation, and other people call it chromosome fusion. This involves at least two breakage, or two double-strand breaks into separate chromosomes. And these breaks here and results to fusion of chromosomes, and which is also a dicentric chromosomes, which is meiotically unstable. And for the cell to make it stable, it has to do some epigenetic changes to silence one of these centromeres to make it kind of stable. But however, underneath this centromere, old or the ancient centromere, if you look at here, it's also highly repetitive. So when we do fluorescence in situ hybridization to understand, to check the DNA sequences here, we could still see satellite DNA is present here, although this is no longer functioning as a centromere. So in a way, if we use cytogenetics, we can see if we can tell that if we find some of these kind of old centromeric repeats found in these chromosome arms, this could tell us that this has been a product of chromosome fusion. So in a way, this could be a diagnostic, it's like a relic of ancient fusion, and could diagnose if we compared it to their parents. So the more patterns of these could tell us more events of chromosome fusions. Essentially, this is simplified, but there are many things going on in evolution. But I want to just emphasize this, because we will come back to this later on in the presentation. And several works and have really confirmed this, that in different these species here with low chromosome counts, and in different assemblies, they have found that fusion or junction in chromosome blocks are actually flanked by these different satellite repeats, which could be, in this case, satellite DNA, ribosomal DNA, or telomeric DNA. So I hope I have given you just a very, very quick tour about the relationship between whole genome duplication, repeat burst, diploidization, and how these repeat bursts influence chromosome change and speciation, and in turn, and this cycle. So now I want to introduce you to the star of this work, which is the crocus. The crocus belongs to the Iridaceae family. It has relatively not so many species, 240, but it is very rich in dysploids. We have about 20, or there are so many different chromosome counts in this genus, and these are just a few of the representative species here with Oliveri with chromosome count of 6 and up to 64 in Carpetanus. And if we compare this with Ploidy, we are curious why in this subfamily level, it has essentially these Ploids maintained the basic chromosome number of 7 without really changing to different basic chromosome count. While in Crocus, in this genus level, we have seen so many. So essentially, we are wondering maybe there are some factors, which is one of the things that we want to explore in this future studies as well. But for now, we think that Crocus is an exciting genus to study dysploidy or change in chromosome count. So we cannot just study the whole genus altogether at once, because this will really be a very big work. So we focus our work first on this particular group here, series Verni, or we call them the spring crocuses. Not just because we have enough genomic data or available sequencing data, but we also because this series is essentially very rich in dysploids. Like each species is essential having this all different chromosome count. Like we have here 8, 10, 12, 14, diploid 16, and 4. We have some tetraploids here as well. And Crocus longiflorus, which is sister to the core Verni group, has very small genome size, and but the highest chromosome number. And if we plot the chromosome number to genome size, they have very negative correlation, which tells us somehow that this is dysploid. Also, just a quick overview of their distribution. The Verni, particular series Verni, is distributed from the Pyrenees to from the west to the Carpathians to the east, and southern Italy. And they like to they thrive very well, and they're very in the cold, in alpine environment, and they are one of the early spring flowering plants, and they're very pretty. One nice thing about crocuses too. And so we focused on this, and we asked ourselves if whole genome duplication promote dysploidy and species diversification in crocus. So this is a kind of broad question. So we want to hypothesize that this whole genome duplication promoted dysploidy, but we want to incorporate the role of repeats in this process. So we believe that repeats played a big role in the increase in dysploidy in crocuses. So we just narrowed down our work our work for this time. We want to explore just the relationships between whole genome duplication, repeat dynamics, and dysploidy, and in this particular series, Verni. But in order to do this, we need to, of course, identify whole genome duplications and map them in the phylogenetic tree. And we need to design or identify all the repeats in all the accessions that we study, and want to understand how these repeats, maybe find some hints or clues how these repeats influence or has a relationship into the change in chromosome number. And so to do that, we had to assemble the genomes but we are limited financially, so we just have we use transcriptome and genomic data to calculate the KS analysis and find out the whole genome duplication events. And with this, we could tell that there are two main events, whole genome duplication events in Crocus, what we term as Crocus or CR beta and Crocus alpha. The beta being the older one prior to the diversification of Crocus around 17 to 19 million years ago. And the second more recent one is that is dividing the series or before the diversification of each series. And we have particularly here two clear genome assemblies for series Crocus and series Verni, and we could plot them around this time, 3.7 million years ago. And since we focus on our series or series Verni or spring crocuses, I just want to highlight here that this whole genome duplication is not shared by Crocus longiflorus, which is kind of at the root of this group. But after the diversification and after the split of this core Verni, and only this group here experienced whole genome duplication. And with that, we want to ask now, because that we have at least for these two series, we could really see that there are whole genome duplications. So we want to ask, are there coincidental repeat bursts, or can we see repeat group specific amplification of repeats, especially for this series Verni? So we did a panrepeatome analysis. We identified or analyzed all repeats using Repeat Explorer to analyze the repeats in each 219 accessions that we used. And with that, we could see here on the x-axis are the group-specific repeats. So these are the significantly associated repeats to this specific series. And on the y-axis here are the 219 accessions. And we can clearly see that at least for the series Crocus, where we have whole genome duplication data that we could confirm, and as well as for Verni, we could really see certain type of repeats that are highly amplified here, as well as in Verni, but we couldn't, they're very low or almost not present here in the other groups. And the same with other groups. So although we have whole genome duplication data only for series Crocus and Verni, we could kind of extrapolate this to say that perhaps all these other series experience whole genome duplications prior to their diversification as well. But particularly in this group, we believe this is a very messy group. We believe that there's a lot of integration, hybridization, and other things going on here. And using these repeats, group-specific repeats, we could also clearly see them, the grouping in this PCA plot. So now that we have these repeat information, we want to ask, can we use this repeat information to understand dysploidy in Verni? Because our initial question is the relationship between whole genome duplication, repeat dynamics, and dysploidy, change in chromosome count. Now we have, with this, we have already seen that whole genome duplications, at least with our available data, could see from Crocus and Verni that they had whole genome duplications, and then these whole genome duplications potentially caused or promoted the amplification of these particular groups of repeats here, which we think are similar to the other series here, but we don't have data to do that. Now, what we want to know more is that how are these, or can we see the involvement of these repeats to really influence the change in chromosome number? Because essentially that is what we want to know. If there's a change in chromosome number, and eventually this chromosome number, could they kind of influence speciation in the future? Oh, yeah, eventually. So to do that, we use satellite markers. because satellite markers also show group-specific amplification. Like for this Adami group here, we can see that this particular satellite repeat is highly amplified here, and also in this punctati group. And you can see a pattern which is also showing group specificity. And so with Verney, we just selected these 3 cytogenetic markers because satellites are also, like in the introduction I mentioned earlier, that they also show really nice distribution in the chromosomes, so we can easily watch them and maybe see them how they move around the different species. So we designed cytogenetic markers for this. And by the way, this pattern shows us these are satellite markers based in the repeat explorer. So we designed probes, fish probes, and we did a fish analysis. And these are the 3 satellite markers that we identified. And we also use the commonly used universal 5S RDNA and 35S RDNA markers. And by the way, Crocus has a different telomeric repeat from Arabidopsis, and Crocus has a human type instead of the Arabidopsis canonical plant telomere. But I want to bring your attention here. The Crosat 114 is essentially present in few chromosomes. If you look at here, they're also present in few chromosomes, but essentially very kind of random and not so no specific location, but they're interstitial. And this red one, Crosat 137, is centromeric, and Crosat 42 is more interesting because they are found in the telomeric area, subtelomeric, and the centromeric, some parts, and interstitial, so they're all over the place. And I just want to focus your attention here in this pink Crosat 42, because if you look at here in Longiflorus, remember this Longiflorus is the sister which did not experience whole genome duplication. It has, which also has the smallest chromosomes and still the most abundant chromosome compared to the others that experience whole genome duplication. And those that experience whole genome duplication, you can see them, that the fewer the chromosomes become, also the bigger their loci of this pink, and also the more loci occur. So this brings us to this introduction I mentioned to you earlier, that this could be landmarks or kind of diagnostic feature of chromosome fusion. Of course, this has been hypothesized, but we could see them cytogenetically. And just to make it clearer here, maybe the other slides or the chromosomes are tiny. We can see here, and this based on this plot, we plotted the number of loci and also the abundance, the total accumulative length in terms of megabase pairs of these different repeats, and we plotted them against the chromosome count. If you look at the 42, it's pretty, there's no relationship regardless of few chromosomes or many chromosomes. There are essentially a similar count and abundance in the genomes. And this red here, which is centromeric, essentially it makes sense because they are centromeric. The more chromosomes you have, the more loci you have, and also the more copy numbers you have, as you can see here. Yeah. But interestingly, this Crosat 42 has a very negative, strong negative correlation. So the more chromosomes, the species with more chromosomes have very few and very low abundance compared to those that have very few chromosomes. And you can see here clearly in this ideogram, Crocus longiflorus, which did not experience this whole genome duplication, has only one copy or one chromosome bearing this satellite repeat here at the subtelomeric repeats of chromosome number 7. However, other species that after the whole genome duplication event, as they change or as they fuse chromosomes, the number and the loci size become bigger, and also they have become more abundant. So we wanted to see maybe to confirm, we hypothesized that after whole genome duplication, as I mentioned to you, this repeat burst and the chromosomes fused, we hypothesized that these chromosomes here in dysploid chromosomes from species with fewer chromosomes are results of fused chromosomes from this longiflorus, the sister to this Corverny. So and that we also hypothesized that these repeats flanked these different chromosomes. So for example, maybe this chromosome 9 or this region here of this chromosome of Vernus, Crocus Vernus, maybe is one of the chromosomes here. And then maybe this other fragment here, block here, maybe is representing another chromosome here. So we think that these are composed of the same chromosomes of longiflorus, but are fused in a very complex way. And that this pink CROSAT42 facilitated this fusion because of this ectopic recombination, homology mediated recombination. But to do that, we need to design a way to understand this fusion here. So we had to assemble the genome of longiflorus, and then we designed chromosome panes, chromosome arm specific panes, 6 chromosome arm specific panes. And when we performed fluorescence in situ hybridization, we could confirm that Crocus longifloris is a diploid, that they have really 2 good pairing of chromosomes reflecting the pairing here, and chromosome 11 here, 13 green, 14, and number 3 here as white. So yeah, they're exactly confirming what we have in our assembly. And but the interesting thing here is when we use the same fish probe, chromosome painting probes to Crocus Vernus. Maybe this is a little many signals you can find here, but I want to take your attention here. This is the chromosome paint. We can see clearly that in one chromosome, so we have the orange, the yellow, and the green, but not in one chromosome. But not only that, we could also see many recombinations or like reshuffling of the chromosomes in this Crocus Vernus. Like in this, you have orange in the ends, but after in this one chromosome here on the left, it's yellow and then green, but here is purple and yellow. And here it's yellow, purple, and yellow, and here it's only blue. And it's just all over the place, reorganized. So we could, with this result, we could really confirm that the chromosomes in Crocus Vernus really increased, or the genome of Crocus Vernus increased, and also the repeat number and abundance increased. And this is also concomitant with the fusion of different chromosomes from longiflorus. And we also could see that CROSAT42 is flanking these chromosome blocks. So with this, I have shown you that the genus Crocus experienced at least 2 rounds of whole genome duplications, and more recent is Crocus alpha and Crocus beta, the older one. And I have also shown you that repeat bursts, we could really see repeat bursts, which is coincidental to at least the Crocus verni and Crocus rehab genome data, which we think some other series are also experience their respective whole genome duplications. And we could, I have shown you that CROSAT42 potentially facilitated descending dysploidy in series where for now we only have cytogenetic data, but we are now sequencing the genome, and we want to get more information and hopefully get the mechanisms how this repeats really in action is involved in this whole genome duplications and dysploidy. Maybe some stress and some proteins are facilitating this, and so this is one direction of our research we want to explore. And I have also shown you that chromosome oligopanes is really useful to visualize the chromosome fusion in Crocus Vernus. And lastly, I have at least hopefully shown you the relationship between whole genome duplication and that this whole genome duplication triggered or promoted repeat burst. And this repeat burst, at least not all of them, part of some of the repeat bursts may have influenced or actively taken part in the change in chromosome number in Vernie. So with that, I would thank your attention, and I would like to thank our collaborators in our group, Dorta Harpka and Frank Plattner. They were the one who made this proposal, and they hired me as their postdoc, and our colleagues here who helped us also with chromosome counts and stuff, Andreas Huben and Ingo Schubert for critical discussions, and our colleagues for other technical and sequencing stuff. Thank you so much. I'll be happy to welcome some questions.
All right. Well, thank you for another wonderful talk, Nomar. If anybody has any questions, please feel free to raise your hand or put your question in the chat. Well, I'll ask a really quick question that sort of I had during your talk. So for the different these satellite repeats, the SAT42 and the other one, how different are they? Are they I didn't are they the sequences themselves? What are the size of those satellites, and how different are those sequence compositions of those satellites? Does this, yeah, and then sort of just think about where those are they just being co-opted as a part of that fusion process, or yeah. Anyway, what are your
They're
yeah
very different.
yeah
Sequence-wise, they're totally different, and also their lengths are also
there you
vary.
go. Okay.
Yeah, and they don't really share any portions with each other. So I think they have very different evolutionary pathways, how they amplified. But my one of the questions we are also curious in answering is how the genome or the genome choose a certain repeat to amplify, and from here there's
Right.
only copy, one chromosome, and in here they become so many. How did from here they become so many loci? So there must be a mechanism of a kind of structure in the cell that use machinery that use these repeats, but how they choose that, it's
Right. What sort of coevolution with some other structure or something, right? That is attaching to those hypothetically, or chromatin or something, right? Yeah, that's interesting.
right.
Yeah.
Yeah.
Oh, very cool. Thank you. Yeah, I missed the lengths. I was looking at all the other parts of their figures. Beautiful slides.
Thank you. Yeah, so this is a question we want to explore. This is a 6-year project, and I'm just finishing the third year, and hopefully we get the extension, and I want to learn more about the mechanism, really what's going on inside the cell.
Yeah, it's really wonderful. Any questions from any other folks right now for Nomar or for Maya? All right. Well, it's a quiet day. Well, I appreciate everyone. I appreciate both you and Maya giving us two really wonderful talks. These are super interesting and exciting to see the research that you both are doing. And yeah, no, thank you again for coming and speaking to all of us today. I just want to highlight that our next Polyploidy webinar will be on April 2nd, the first Wednesday of April. And we'll have Raymond Castillo and Eric Hagen speaking then, and that'll be our last Polyploid webinar of this semester. We'll start back up in the fall. And if anybody's interested in speaking in that, then feel free to shoot an email and look out for soon enough. I'll send around a sign up for everybody to start signing up again for next fall. But yeah, I appreciate both Nomar and Maya for speaking to us today. Thank you so much.
Thank you
I'll
for the
post
opportunity.
absolutely. All right. Well, everyone have a great afternoon or a great evening, and we'll see you soon. Thank you. Bye.